Ever wonder how the food you eat, like pizza, turns into energy? It's all thanks to cellular respiration. Imagine a tiny power plant inside each of your cells working tirelessly to convert the food you eat in a form of energy our body can use. Cellular respiration is like a chain reaction, breaking down the food molecules and releasing energy in small, manageable pockets called ATP. ATP is like the body's currency, powering everything from your heartbeat to your brain power. Where does energy really come from? Technically, it arrives from the sun. The sun is releasing huge amounts of energy as a byproduct of nuclear fusion reactions, and the sun energy comes to Earth, where it can be absorbed by plants. Plants use sunlight, carbon dioxide, and water to make glucose, a type of sugar or their food. And all of this glucose and other biomolecules that becomes the starting material of metabolic processes in our bodies. This degradation of biomolecules to generate energy that cells can use is called cellular respiration, which is what we will be discussing about. What is cellular respiration? Well, let me break it down for you. The cellular respiration is a process to make energy. This is the process by which cells convert glucose into energy. This process is located into the mitochondria. Mitochondria is famously known as the powerhouse of the cell. Primary function is to produce energy in the form of adenosine triphosphate or ATP. Cellular respiration consists of three main stages. 1. Glycolysis. The first step, glycolysis, occurs in the cytoplasm of the cell. Here, one molecule of glucose, a 6-carbon sugar, is broken down into 3-carbon molecule called pyruvate. This process does not require oxygen and produces a net gain of 2 ATP molecules. Additionally, it generates 2 NADH molecules, which are used later to carry electrons in the final stage. The next stage is the Krebs cycle or the citric acid cycle. After glycolysis, the pyruvate molecules enter the mitochondria, where the citric acid cycle takes place in the mitochondrial matrix. Before entering the cycle, pyruvate is converted into acetyl-CoA. The Krebs cycle breaks down acetyl-CoA, releasing carbon dioxide as a waste product. More importantly, it produces high-energy electron carriers, specifically 6 NADH and 2 FADH2 molecules, as well as 2 additional ATP molecules per glucose molecule. These electron carriers are crucial for the next stage. We are now in our third stage, electron transport chain and oxidative phosphorylation. The final step is the electron transport chain ETC, which occurs in the inner mitochondrial membrane. In this stage, the electron of NADH and FADH2 produced in earlier steps are passed through a series of protein complexes in a series of redox reactions. During this reaction, NADH and FADH2 are oxidized as they lose electrons, while the protein complexes are reduced as they gain them. This movement of electrons generates a proton gradient across the membrane which powers ATP synthase, an enzyme that produces ATP. Oxygen is essential in this step as it acts as the final electron acceptor and is reduced to form water by combining with electron and protons. The ATC is the most energy efficient stage, producing approximately 32 to 34 ATP molecules per glucose molecule. In summary, cellular respiration converts one glucose molecule to approximately 36 to 38 ATP molecules. It is a critical process for providing energy to cells, enabling them to perform essential functions such as growth, repair, and maintenance. Now that we cleared that, let's proceed to the topic deeper. Imagine powering a machine that needs energy to work, but what happens when oxygen isn't around? The machine switches to fermentation, which gives energy but not as much. It converts glucose into either lactic acid or ethanol and CO2 depending on the type of fermentation. Want to know a cool trick? Your machine can also break down fatty acids through a process called beta oxidation, turning them into ATP. And here's a key point. NAD plus and FAD help out by oxidizing, losing electrons, and passing them to the electron transport chain (ETC), where most of the energy is harvested. But there's a catch. The machine has to make sure that it's not running empty, so it uses feedback mechanisms to regulate energy reduction. If it has too much ATP, it slows down to avoid wasting energy. Also, this process doesn't just make energy, it also helps in building important molecules through biosynthesis. Also, this process doesn't just make energy. It also helps in building important molecules through biosynthesis. And the best part? It's flexible. If carbs aren't available, it can switch to breaking down fats or proteins to get energy. Pretty versatile, right? But wait, I have a little joke. Why did the mitochondria get a promotion? Because it is a powerhouse of the cell. Speaking of powerhouses, aerobic respiration is the real MVP, generating more ATP than its anaerobic counterpart. Anaerobic respiration is like that friend who always shows up late and only brings half the snacks. It gets the job done but not as efficiently. You journey from high energy efficiency of aerobic respiration fueled by oxygen and producing copious amounts of ATP to the less efficient but essential back of anaerobic respiration. We've seen how crucial molecules like NAD and FAD act as electron carriers, delivering good to the electron transport chain, the cell's ultimately energy factory. And we've marveled at the body's sophisticatedly regulatory mechanisms, ensuring energy production is finely tuned to meet the cell's need. Ultimately, cellular respiration isn't about energy. It's dynamic, adaptable system that underpins life itself, seamlessly switching between fuel sources and pathways to keep the cellular machine humming along. It's a testament to the incredible complexity and elegance of biochemical processes. Thank you for watching my video. Before you leave, click like and subscribe. Thank you.